Thank you. And thank you to all the speakers once again who are joining me here on our virtual stage for our rousing Q&A. I know that we only have just about 30 minutes here. And so I actually want to kick off this Q&A with a question that sort of has been floating in the back of my mind as all of you are mentioning this one specific type of bacteria, lactobacillus, right? I think that has been a common thread throughout all of these talks today. And I'm just curious in terms of what you think in terms of actual clinical implications and what we can be doing in the clinic to sort of translate some of this research to what we're seeing in the clinic. I know that there's a lot of implications about lactobacillus and what it's doing, but should we be having our patients place lactobacillus suppositories? Should we be telling our patients they need to be eating more yogurt with lactobacillus? What do you think is the key here? What should we be doing with this information right now, if anything? And I'll lock that to any of you.
I mean, since I just talked kind of about lactobacillus, I comment from the bacterial vaginosis standpoint. So nothing has been, our silver bullet here, but there are some trials that have happened. So like MVIF30, for example, was a big trial that they did with lactobacillus suppository. And, there is some efficacy, especially mixed with antibiotics, but I don't think anything's good enough. And there's been a lot of evidence that orally taking lactobacillus has not had quite the effect that we'd like to see it in the vagina. Again, there's a lot of controversy in the different studies. There's also, I want to just point out, a lot of products on market that say, women's health, probiotics for, the vagina. And then you look at it and it's like lactobacillus GG or lactobacillus plantarum. These are gut bacteria that don't even make lactic acid. So it's not going to have any effect on the vagina for you. I always say that, it doesn't hurt to take probiotics, but I don't think it's going to be what, fixes things here. And I think we need a multi-pronged approach as far as bacterial vaginosis goes. And I'll stop there and let the rest of the investigators comment.
Anybody else want to chime in? Thoughts on lactobacillus?
Well,
go ahead.
No, go ahead, Katie.
I was just going to say, this is why we've been trying to advance the experimental model systems, is to actually see, because the human studies that have been shown today and that we're referencing from the literature are beautiful, but there's still this element of association. We're still sequencing, we're seeing associations with health or recurrence or persistence. And we're not actually knowing, is the host driving a lactobacillus dominant state? And is that what we need? So the Lactin-B trial that came up, it was effective in, I think, 30-ish% of women, but just introducing Lactobacillus crispatus alone isn't sufficient to achieve the Lactobacillus crispatus dominant community or achieve resistance to recurrent vaginosis or recurrent urinary tract infection. And so there's still this element of, we don't yet know. I mean, lactobacillus is great. It doesn't have any virulence factors. It produces this lactic acid, which what we've seen in vitro is that really drops the pH low. And it's once that pH gets dropped, I mean, the ability of these other organisms to grow is really deficient. And so that might be its main mechanism, is that it doesn't have virulence. It drops the pH and prevents overgrowth, and then it's immunosuppressive. And the culmination of that is what you need to get a healthy urogenital microbiota. So, I guess I'll stop there. But I think it's beyond a single organism. It's this composition or this activity that we need, and a host state that permits that activity that we need to actually improve the health.
Yeah, I'll just say people who are affected with chronic vaginitis, including chronic BV, with recurrent infections in the urinary tract, when they're doctors and when science doesn't give them something that solves a problem, I mean, these are not stupid people. They're going to try to figure out something. And we shouldn't laugh at the fact that they do these things that sound really goofy to us. But I think we can discourage people from putting yogurt in their vagina and things that are really have no chance, but they might sound good on a throwaway women's magazine at the grocery store, right? Because that's a wasted effort. That's just going to, that's not going to help them. It's not going to harm them, but it just is not moving toward health. So I just talk about the limitations of the evidence And I encourage people to follow a whole food plant-based diet with the goal of trying to at least populate their gut microbiome with a more beneficial microbial community, assuming that that may help other nearby niches have better communities. Can't prove that, don't know that. But to try to, like, in the urinary tract, people are like going over the top with cranberry, for example, right? They're going to turn into a cranberry. They're taking so much of it. It doesn't make any sense. And so that, and the yogurt and all that stuff, it's not helpful. And we need to recommit to trying to get helpful answers to affected patients, and try to gently talk with them about, we're sorry we haven't come up with anything better, but we're really sure that putting yogurt in your vagina is not going to solve the problem. So please don't do that.
Yeah, I will add that, we do know now that there's, lactobacillus dominans is not universal in healthy women who have no gynecological or obstetric issues. And that's particularly true for women who are not white. A lot of studies have been done predominantly in white communities, and that was seen as normal and healthy, and everything else as something, that was worrying potentially, which may not necessarily be the case. I think as Katie was saying, I think it's potentially have to do more with the environment that is created by these microorganisms, which is not exclusive to lactobacillus. There are other species that can do these things, which are basically bacterial warfare that create an acidic pH that makes it very difficult for a lot of pathogens to grow. So, just the fact that a particular woman doesn't have a lactobacillus dominans, it doesn't mean they need to try to fix something. If they have no symptoms and no issues, I would say that's really not encouraged necessarily, because they may start having symptoms after they try to do something about it. And also the probiotics, I would say it's not necessarily true that they're not harmful. They can be. Everything we do can be harmful, right? So I think it's always worth thinking about everything you do in your lifestyle choices and everything, because all of it comes with a risk. We don't understand that very well yet. I don't think any of us do yet, and we're trying to understand it and help all of us make these decisions. But nothing is risk-free, including those things. So just something to keep in mind. If you don't have any reason to try to fix yourself, experimenting with these things can cause problems. It's possible. So just be cautious, I would say.
I just wanted to add one other thing. We think of lactobacillus makes lactic acid, and we're all happy about that, lowers the pH, all that good stuff. But there's a cell-free supernatant story that I think can help also with bacterial inhibition, sort of the bacterial warfare that Marina is talking about. So it's got talents that we don't have fully elucidated just yet. And how and when it brings those talents out may depend on what else is going on in that community. And I don't think we have a full understanding of that just yet. So my goals for lactobacillus is, if it lives there, don't try to wipe it out, like with excessive antibiotics, speaking to you doctors. And, don't do wacky things to try to get it to grow there if, we don't have evidence that that's going to be effective. Those would be my 2 rules for lactobacillus.
That's a great point. Thank you all for contributing to my probing discussion about lactobacillus. We'll go with in order, I guess, here. There are a lot of questions in the chat here, so we'll start with Dr. Patras here. One of the people here joining us today asked, can you expand on the simulated vaginal fluid you're using?
Yeah, so we adapted a method, I think I had the citation in my presentation, of a simulated vaginal fluid that was based on proteomic analysis of human vaginal fluid. So it's pretty nutrient poor. It has glycogen, glucose, urea as a nitrogen source, peptone as the amino acid source, mucins, lactic acid. That was our base media. And then we've been adding host components. So we've tried things like tween, what should be a fatty acid supplement. We've tried blood as a nutrient to help promote some of those more fastidious organisms. We've incorporated vaginal epithelial cell lysate, which would also make sense for some of these organisms that are a little bit more host adapted. And so, yeah, so it's kind of a a shift from what's been published, and so we've tried a bunch of different conditions, and so right now, it's primarily mirroring the vaginal fluid based on published studies, and then we've started to add some additional components that mirror the host, because that's what we think is missing in some of those studies. The normal simulated vaginal fluid that we started with grows, a lot of E. coli and staph and all those things that are really fast growing, but to get some things like Gardnerella or Prevotella or Fanny Hessia, we really need to add more of those host components to get successful cultivation.
Have you tried adding any therapeutics to that, like a vaginal, like estrogen, to see if that brings in any lactobacillus?
So estrogen is an interesting point, and that's I'm sorry, Dr. Brubaker has thoughts on that as well. So that's where I'm getting at with more of this. I think we need to take this more comprehensive picture. So that the success of vaginal estrogen, estrogen is such a potent stimulus for a variety of different cell types, and so that's something where we need to understand more of potentially the mechanisms creating a healthy associated host state. If it's a metabolite that might be beneficial, that's more towards where we need to go. We haven't yet added steroid hormones to our bioreactors. That's still not necessarily modeling the host. I told you we're adding host components, but there's still no host cells themselves in that. We also haven't yet added them to the organoids. There's a lot of growth factors that get proliferation of those organoids, and so estrogen might not be as needed in those cases, but that's certainly something down the road that we could look at, and certainly something that would play a role in immune modulation as well, but not yet, not estrogen.
Great, thank you for that. Next question I will toss up to Dr. Walther-Antonio here. It says, what is the current status of microbiome-based or aided endometrial cancer diagnosis? So is this something that can be commonly checked, or is this something that is asked for by patients?
It is something that, I do receive emails about fairly often, because, it's published work, but it's still in the research realm, right? So as I mentioned at the beginning, that was in my disclosure. This is being explored by some commercial partners that Mayo Ventures is partnered with, and be part of clinical trials and things like that. So it's something that it's in the pipeline, and I wish that we could do this in tomorrow and help patients. We cannot say test it and then figure out. We don't know if this is a real thing, right? There's not enough patients, there's not enough diversity in our population to really for this to be ready to be a clinical test with meaningful results that people can trust and act upon. So we are still investigating. Hopefully it'll be something in the future will be helpful.
Great, thank you. Dr. Brubaker, I'll toss the next question to you. It says, thank you for sharing your research. At-home sampling, as you've shown, is much easier for patients, and you touched on this a little bit, how can you assure sampling quality and accuracy? Any strategy you're using to preserve the DNA or minimize contamination?
So in this study for the PLUS RISE study, we thought about that very carefully, and that's one of the things we wanted to test specifically in this feasibility study. That manuscript's just been accepted. The methods for all these, the larger study and the feasibility study, are out, and so you can pull those up at any time. And so we use the PZ device to reduce the risk of contamination from a voided specimen. We consider that a urogenital specimen, according to our consensus document. And then we had participants pour it into a tube that had Assay Assure, a DNA stabilizer in it, before shipping it in a prepared kit via FedEx, where it went to a staging center before it came to the lab. So all the methods of how we did that are in a neurourology urodynamics article last year, I think. And so we took those steps, and we found that it was sufficient. And importantly, we found that just because somebody is in the research center, it doesn't magically all get better. There's challenges to collecting a low biomass voided specimen, no question about it. So you have to factor in all those things very carefully. And then there's analytic issues that you have to be very careful. Someone else has to question about that, and how do you work with that low biomass specimen? And you have to just throw away a bunch of sequences because they're below the sequencing threshold that you set a priori.
Great. Thanks, Dr. Brubaker, for that helpful tip on studying low biomass. As somebody who's struggled with that in his own research, I can definitely empathize with that. Next question I will ask to Dr. Klatt here. And this is interesting because it's sort of been asked like a little bit of everybody here, but any possibility of something like fecal microbiome transplant-like therapy for the vaginal microbiome to prevent recurrent BV?
Yeah, I'm so glad this was actually asked, because after I started talking about the probiotics earlier, I thought, oh shoot, I should have brought this up. So thank you for whoever asked this. So there was a really nice study a few years ago by Aran Elenov's group, and they demonstrated that there can be some efficacy with vaginal material transplants. And so it's a concept that is often talked about. The big problem is, just like a fecal transplant, you always worry about something that you don't want to get in coming across, right? And so there's been a lot of work done in optimizing that in the context of a fecal transplant, but as usual, not as much work optimizing anything for the vagina. So I think that it is an option, as a lot of people as all these women here today have touched on, right? Like, it's probably more than just that one bacteria. But what's making that community structure? What metabolites are there? What mucins are there? How is it interacting with the immunity and that barrier? So the concept of doing a actual, fecal material or vaginal material transplant, is definitely enticing. And like I said, there is some data to support it. That being said, there have been no, and correct me if I'm wrong, anyone, but that I'm aware of, placebo-controlled studies to actually look at this and determine the safety and efficacy and long-term effects of this. But it's one of the many things that I think and my wheels were spinning as Dr. Patras was talking about the modified kind of her communities, and trying to do something together with that to make something that is sort of a synthetic transplant, if you will. So something like that, I think, again, a combination approach is what we're going to need.
So that's interesting that you mentioned that, because I think I have another question that I'd like to actually ask the group at large, because it's kind of a theme that has come through in some of the questions here, especially for those who engage in human subjects research. I think one of the things that we touched on is not only engaging in community partnerships and how we do that well, but also the importance of increasing the diversity of and representation from underrepresented, underserved, historically underrepresented minority groups in research. And so I'm curious, research, as we're all speaking about this, and everyone has put up all this like multitude of collaborators that they work with. How do you do that? How is it that you engage with communities? How do you diversify your recruitment efforts for the trials that you publish? What are some tips and tricks that you have to share with the audience?
I'm happy to talk a little bit about, in the PLUS consortium, we have a group, we actually have a community advisory board, and we have rapid assessment partners, so that each clinical center has their relationships with community members. And so they've been involved for the nearly 10 years that this research consortium has been up and running. And then specifically, when we did our surveys, we did a formal translation of all the instruments into Spanish. And we did not only linguistic acculturation, but also culturally adapt the changes. So those instruments this is taxpayer dollars, so that will all be publicly available. And then with each wave of the survey, with each replicate of the survey, we matched against what we wanted to get to mimic the US population. And then we adjusted our invitations so that over time we had the age and racial and ethnic distribution that we wanted. And so that then came through in our larger circle of all the participants, those people who came for the in-person visit, and those who participated in the feasibility replicate of the biospecimens. And it's not, to 3 decimals or anything like that, but it's pretty darn close. And so by being very intentional about it and having a commitment to that kind of effort, it just really is infused through our entire research consortium. And nothing happens without that, including the lay summaries that we do for all the research papers that we do, or nearly all the research, so that it's in simple language, common language summaries, so that we can get the information back to communities and the research partners in the community.
Great, thanks for that, Dr. Brubaker. That intention is certainly really important. And lay language is, as we know, really important for the dissemination of the work that we do.
Yeah, and I will add that I do find this quite challenging often, and it's most of the time, or almost 100% of the time, it has nothing to do with the communities and a lot to do with just how we do science, how we fund science. That's what the barrier, I would say, mainly is. It really can break trust between the people and those conducting science. In our case, we're trying to engage with not just the patients, but to enroll in our studies, because we're interested in not just what's happening to the patient and the host, but also the environment. And so it's very important that we actually collect samples from the household, and for a period of time that involves years at a time. So to really have that connection with them, that they trust that we're doing what is appropriate with those samples, and often the conversations we have with them is really even asking a simple question like, what do you think is happening to you? What do you think? Nobody ever asks them that. It's always, we tell them what's going on with them, and don't hear as much as they have to hear us. And I think that that's something that needs to change. It's just finding resources. This is very resource intensive to build these bridges and maintain them. And often that's very difficult to fund, and it takes time. And as Linda was saying, these efforts, not just ours, but others. I was just at the Translational Sciences meeting, national meeting, and on average, it takes 10 years for these things to go anywhere, and be ready to deploy with some security. So it's what kind of funding mechanism, right, that we have currently is going to support that sort of thing. It's always with several sources here and there, and it just makes it very difficult for everyone. I wish there was better infrastructure to support these efforts in a more sustainable manner that makes it easier, not harder, for people who want to do it.
Yeah, and if I can comment on that too, I do think this is a major problem and why we are so behind in the field. For sake of time, I didn't have a slide of how many treatments we have for bacterial vaginosis versus how many treatments we have for erectile dysfunction. And it's, and one of my favorite sayings is that, if men had discharge and odor and itchiness from their penises, this would have been cured a long time ago. But we are resource limited, and it's very difficult. And we have shown time and time again that different communities have different sexual practices. There's a great question as I was going through the chat. Someone asked about, can BV be because of different sexual practices or increased, promiscuity, which absolutely, we know sexual partners can affect it. But we know things like intervaginal practices and douching and, hygiene practices. And then, you start getting into Africa where when you're during your menstrual cycle, you have to go sit in a hut by yourself in certain populations. And so, highlighting the importance not just of doing this work and doing it internationally and in different settings, which is extremely critical and understudied due to a lot of funding, I think, she nailed it on the head there. But also not just doing, just a biological study. I mean, we need to be partnering, and working with psychologists and people who specialize in behavior and understand these things, and looking at the different multiple facets that actually are going to, play a role in this, because it's not just biology for sure. So, and we do need better ways to address this and better ways to partner and have the funds to do it that, an R1 setting isn't going to allow us usually.
Yeah, I would just jump in and say, in some ways, I feel like I have an advantage because I'm in Houston, Texas, which is reportedly one of the most diverse cities in the world. And so we've certainly been very intentional at trying to reach different communities that are here. So we have both clinics associated with more high income, and then clinics with lower socioeconomic status, like the Harris County health system, which is broadly serving the community. And that's been an approach that we've taken to try to capture some of these underrepresented or undescribed communities that are at most risk for things like type 2 diabetes or autoimmune disease, and to try to, study what the vaginal microbiome might be in those individuals. So it's really important. We're trying. I don't know how perfectly we're doing it, but it's definitely something we're considering.
So much insight and wisdom from this group. I mean, I really appreciate hearing everyone's thoughts about this. I will echo this as what I see day in and day out in my clinic: resources for reproductive health, women's health. It's a call from all of us to help us do these things, to set out with an intentionality, to begin to build trust, to go into these communities, to be able to do research in other places. I mean, I think these are all really important aspects of the research and the work that we're doing. I'd like to shift gears just a little bit. I know we don't have a whole lot of time left, but I think we've talked a lot about none of this just living in isolation, right? I mean, I think we have all of these really wonderful things that happen, but there are host interactions, there are other microbe interactions, there are viruses, there are a lot of things in the body that can potentially modulate some of these interactions. And so one of the first questions that got asked to you, Dr. Patras, is that are there any plans for you to screen for specific proteins or metabolites that help mediate this host microbe or microbe interactions? And is the current in vitro model suitable for compound screening?
Yeah, so great question, and certainly highlights the goal of these models. So a lot of our efforts over the last few years has been to develop and then validate these models that we can stably cultivate complex communities that actually reflect what the input was, and in a way that is as physiologic as possible. So that's why we've made all these different hurdles to try to, adjust for oxygen concentration and flow of our media and various components that we're adding to try to really reflect the physiology. Because even if you have the organisms there, they might not at all be doing what they're doing in the human. And so we've put a lot of efforts in. We're just now getting to metabolomics. We have added not yet, one compound in particular, which is a whole side tangent, and I will get some raised eyebrows if I mention what we're adding. But we've added things like pathogens to see, does the community respond to if we introduce this pathogen? Does it permit pathogen colonization or resistance? Another thing that we're interested in looking at is if we introduce a bacteriophage that has an organism there that it can replicate in, what does that do to the community? So that's where we've started. But yes, the goal would be, of course, to see, if we add a metabolite or something that would be considered a prebiotic, can we shift the community towards either the composition or the function of something that might be associated with health?
Thanks so much for that answer. I'm gonna pose sort of the similar question to Dr. Brubaker, because she mentioned metabolites a little bit in part of her talk. Do you think, Dr. Brubaker, that metabolites are the next step to figuring out, just because it's there, just because it changes with different lower urinary tract disorders, how does that play out? What is the next step? Is it metabolites that we're going to hang our hat on that say like, oh, these derangements and things can potentially lead to X, Y, and Z? What are your thoughts there?
I have a lot of thoughts on it, and I have a lot of excitement at the kind of insights that having a relationship between the metabolome and the microbiome might give us for urinary tract health. We're seeing really, when you look at the gut microbiome, which gets all the spotlights and all the funding and, all the journal articles, you see some really interesting things with metabolome and microbiome connected. And it's going to be much easier to get metabolites out than some of the other work that we've tried to do so far. And so if we're able to begin to kind of work backward from the metabolome to get some insights into the microbiome or the functioning of the urobiome community, I think that will be a really big step forward for us. So I'm looking for that. I'm looking for big insights into how the phages are working there. I think that's another big frontier that we've got to get figured out. So.
Yes, perhaps a lot of these talks and a lot of this research does what research normally does and raises a lot more questions than gives
more research needed. Yeah.
Yeah, more research needed. Dr. Walther-Antonio, I'm going to ask the next question to you, and it's not in the chat, unfortunately, because I'm moderating. I want to ask a question myself. I think, again, this is something that I think of a lot as a clinician and as somebody who has gone through medical training. I think your explanation for estrogen's effect on bacteria, we don't think about this estrogen effect on bacteria specifically, and what's going on with the ability to, act on intracellular iron to then, come up with things like, hypoxia inducible factor and all. I mean, that's just something that wasn't taught to me, or I didn't retain well as a student. There were a lot of chemical pathways going on there. I'm not quite smart enough to follow those. But I will say one of the things that this brought up is we think that endometrial cancer is really mainly estrogen driven. And a lot of things we're thinking, okay, it's mostly driven by obesity. Obesity creates a relative state where there's more estrogen. This was the first time that I thought, oh gosh, I've heard that estrogen actually has these tangible effects on bacteria. My question to you is whether or not you had looked at any samples or specimens of non-endometrial cancer specimens of the uterus that had been exposed to vaginal estrogen therapy, and whether or not you saw the same changes in the bacteria there. Or whether you think it's mediated by the disordered proliferation that happens with endometrial cancer precursors like hyperplasia. I'm just a little curious about that.
Yeah, I mean, I'll start by saying that, and you said that in my introduction. I guess my background is not a medical field at all, right? In astrobiology and missions to Mars and things like that. So, and one of the reasons I made that, I joined Mayo Clinic was because I was studying microbiolites, which is, some microbial structures that form in the bottom of Pavilion Lake, and they are built by unicellular organisms. And when I started looking at the chemical pathways that they use to communicate with each other and form these structures, I started hitting a lot of cancer literature, and that made no sense to me because I was like, well, I'm studying this at the bottom of the lake. Why are they communicating, and why are these pathways highlighting cancer research, right? And that's when I started thinking, this is kind of, close to the beginning of the microbiome field and in the human setting, and start to think, well, if we happen to have these bacteria or these organisms in our body, well, we're not the only ones talking, right? They understand, that there's communication going on here. They utilize the same pathways, same receptors, and they can interfere with all these things, including treatments. So that's where I started thinking about that, and then brought in into the hormonal, background and that hypotheses. And yes, it's something we are exploring. We do co-cultures of the host cells, both benign and cancerous, in the presence of the bacteria and different levels of estrogen and actually progesterone and even testosterone. We are testing that as well because nobody's paying much attention to that, but it's there. So you never know. And so we are looking into impact on transcriptome at large through time in chronic exposures, which is several generations of this. And also epigenome, methylome in particular, we have seen some alterations there as well that are lasting, and then in the animal models that we experiment with. So yes, something we are paying close attention to, and I think to your point, we do think of endometrial cancer as associated with obesity and particularly unopposed estrogen therapy, but that's not necessarily the case for the most aggressive types, right? The type 2s, are women who are often, quite lean and fit, and yet have the worst, cancers, most aggressive and fast progressing and harder to treat. So there's a lot more to understand there than the common, standard interpretation of these things.
Yeah, I guess that's sort of what we find when we dive deeper into all of these things. As Dr. Brubaker pointed out, when we were in medical school, when I was in medical school, we were just taught the urine is sterile, and that's just that was sort of the one slide, and then moving on, that was it. So we'll shift gears a little bit. I'm going to ask this next question to Dr. Klatt, and this brings up some of the challenges inherent in microbiome research. This question says, thanks for your talk. More on Shannon diversity. What is more to maintain a healthy and good responding microbial community, abundances or species count? Any effects from microbial biomass or sequencing depth here?
So, I mean, if we knew that, we probably wouldn't be here, right? So it's great questions and things that we're looking at. Like I said in the very beginning, is that, we really try to focus on not just what is there, but what they're doing. So, I mean, I kind of will say to that all of the above, because I think it's, the players that are there, the activity they have, and then these interactions, there's very, very fine community dynamics in any microbiome, whatever site we're looking at. And we know there's interactions between these bacteria, and then there's crosstalk with immune cells and epithelial cells. So I think that these all go to hand here. And then as far as, the more technical question on biomass and sequencing depth, I mean, absolutely, this comes into play. And we have very stringent thresholds that we use in our lab that we've established for every tissue type or sample type that we use. And Dr. Brubaker had touched on this earlier, I feel for you because urine is probably even harder than vaginal. But I mean, we do skin too, which is the hardest we've done. But it's, trying to make sense of something when you don't have enough community structure to look at it, or you don't have the sequencing depth, or you don't have enough biomass, it's just nonsensical data. So a lot of samples do unfortunately get thrown out because of that. So, I think one of the keys there, back to the clinical side of things, is making sure you're really communicating well with your clinical partners. If you're a PhD like me that has clinical partners doing these things, we talk in depth to our research nurses about how to get samples. And the same thing, we have a short video we give and in multiple languages as far as at-home sequencing or at-home sample collection, and then, we have several different screens on how much DNA is there. Now we're going to pass, QC and our initial steps. And, even after we go, we will have times, and we've done this multiple times, where we get some data back that we have to go back in and sequence deeper because we have to go deeper. So, it's a complex issue. And I think that this is the importance of having thresholds, and there's some published, some not, but, and I think that unifying some of it would be great. But again, sometimes you do what you can with the samples you have in the study you're doing. So it's, but all important factors and all things to think about and assess before starting a study.
Yeah, that's a really great point about doing microbiome research well, is establishing those things beforehand, and the importance of establishing standards that are universal, and the importance for those things. I know that we're running out of time, so I'm going to ask one last question, even though I want to ask way more questions, and I could spend a lot more time with all of you. And it was a question that was posed to Dr. Brubaker initially, but I think it affects all of, I think it affects, all of the research that we're doing here, is really the effect of the menstrual cycle on the various microbiomes that we're studying. Why does the menstrual cycle have that effect? Why do you think that that's occurring? Is it because of the blood that's there, that's an iron-rich environment? Is it the hormonal status? Is it just the day? Is it the time? What do you think about menstruation, the menstrual cycle, as having the various effects on the microbiome?
I'll just say the answer to that is yes, all of the above. But
yeah, all of the above.
You can speak more to this, Dr. Brubaker.
I don't know is the short answer. I don't know what's the cause. All I know is you got to factor that in when you're trying to characterize your research partners and the contributions of the samples they're giving you. You have to understand where they are in their menstrual cycle and their hormonal status to a reasonable degree without making an undue participant burden. But whoever asked that question, could you go get the answer and figure it out and give us a call? That's something we would love to know. Yeah.
It's something that I've always, whenever I've asked that question, even earlier on in my career, got the shrug, and it's like, it's the hormones. It's all the hormones.
Everything in women can't be hormones,
Yeah.
There's biology beyond hormones. That's a news flash, right? So, yeah.
Yeah, and I would say, it's a massive. Think of microbes, right? It's a cataclysmic event to their environment, right? This isn't just a little drop of blood that suddenly shows up. An entire tissue layer falls off, like gone, and is completely reconstructed from scratch, with the presence of stem cells coming from the bone marrow. So it's a massive, massive change. So I would be very shocked if there weren't massive changes in the microbiome. What's interesting, and when you saw the study that we did with the Wellesley college students, what's interesting is how that massive change occurs, and you see it in the microbiome very clearly, that signature, but usually it bounces back. it finds a way back to whatever it was. And that to me is interesting. How I don't know how that happens. Do they stay back and hide for a little bit and then come back, back to the upper layers? I suppose that must be what is happening. But, it is a vulnerable time is what it appears, because from those participants that we had that had shifts, it was precisely at the menstruation that they shifted and weren't able to bounce back. So it does appear to create somewhat of a vulnerable state in the microbiome that could cause shifts potentially. But really how they are able to bounce back or not, it would be very interesting to study. It's a great question.
All right. Well, I just want to thank you all for staying a little bit later than we had planned, and thank you for such a stimulating discussion. I feel like I learned a ton here today, and I feel like I'm so grateful to have been able to learn from you all and this really impressive body of work that you all are doing at your institutions with your various collaborators. So it's important work. We all know that it's important work, that it needs to be funded better, and it needs to continue so that we can come up with answers to a lot of these really important questions that we're asking. So thank you again, Dr. Klatt, Dr. Brubaker, Dr. Patras, and Dr. Walther-Antonio for being here with us today. Really wonderful discussion and really wonderful thought-provoking talks from all of you. Really appreciate it.
Thank you. It's been an honor. Thanks to all my co-panelists. I learned a great deal. It was wonderful to be on a panel with you. Thank you.
I want to email and work with all of you. So thank you.
We'll see each other in 36 hours. We can just pick up where we left off.
We will. We'll have this discussion all over again in 36 hours. For those of you who are staying on for the next portion of MVIF for today, the invited talks will begin at 15 past the hour with a question and answer to follow that. Thanks again for joining our great discussion today. I really appreciate it.
Thank you.
See you all.